Welcome, my wondrous students, to part 2 of our lecture on nucleic acid chemistry. With no fanfare or intro, I'm just going to launch in. As you guys probably already know, DNA contains all of the information needed to code for all of our physical characteristics. Each cell of our bodies contains in its nucleus all of that information. The individual cells don't typically use all of that information, however. Each cell in our body only accesses the information from its DNA that's needed for that specific cell to perform its specific functions. Generally speaking, it leaves the rest of the DNA alone. But it does have all of the same DNA that all of the other cells in our bodies have. Hence, every cell in our bodies contains all of the genetic information necessary to code for all of our physical traits. So how are physical traits passed on from one cell to later copies of that cell? Well, it's through DNA replication. I'll save the details for our later biochemistry class, and I'm sure that you'll learn even more details if you take genetics. But I'll just tell you that every time our cells have to copy themselves as we grow, they do so by mitosis. Each time a cell makes a copy of itself, it has to copy all of its DNA. How does this happen? Well, when the DNA needs to be copied, enzymes open up the cell's nucleosomes and histones inside the chromosomes and expose that DNA. The two complementary DNA strands are then separated, and the cell's enzymes begin copying each of the old strands, called parent strands of DNA. This process is called replication. Now you know, or should know, the DNA can only be copied in the 5' to 3' direction. Because of this, only the daughter strand on the left can be synthesized in a continuous single piece. And the reason is because it is being synthesized in the 5' to 3' direction. You'll notice that the parent strand on the left is being read in the 3' to 5' direction, which is complementary to the daughter strand being synthesized in the 5' to 3' direction. Now, because the daughter strand on the right over here, you'll see that it's written in this sort of lighter blue shade, is running in the 3' to 5' direction. And remember, it can't be copied that way. It can only be copied in the 5' to 3' direction. Because it's running in the wrong direction, it actually has to be synthesized in discontinuous fragments from the 5' to 3' direction down. Does that sort of make sense? So it gets synthesized in discontinuous fragments from the top down, 5' to 3'. And then once it's synthesized, the individual fragments are joined together by an enzyme called DNA ligase. When the cell's entire genome has been copied, that is, all of its DNA, the two new double helices, composed of one of the original parent cells together with a new copied daughter strand, and then the same over here to the right, then separate, and they go into the two dividing nuclei that are formed when the replicating cell divides. Here's a cool link to a video produced by freesciencelectures.com. I don't want you to worry about all of the more advanced terms that they discuss here, like lagging strand or Okazaki fragments, which I haven't introduced to you yet. Just soak in the wonder of the molecular machinery found in all living cells, as well as the super cool sci-fi techno music.
When DNA replicates, its strands are separated by the enzyme helicase. Single-stranded DNA binding proteins keep the strands from re-annealing. One DNA strand encodes the leading strand, which forms from its 5' to its 3' end, using DNA polymerase III. No problem here, but the lagging strand presents problems. It has to form from 5' to 3' too. It forms in pieces called Okazaki fragments. First, an RNA primase lays down an RNA primer. Then, DNA polymerase III lays down new DNA. The process repeats again and again. DNA polymerase I replaces the RNA primers with DNA. Finally, DNA ligase links the Okazaki fragments.
Now here is another cool video that shows the same thing done with slightly better, albeit faster, animation.
Using computer animation based on the latest research, we are now able to see how DNA is actually copied in living cells. You are looking at an assembly line of amazing miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into two strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is two new DNA molecules.
I hope you find this as amazing as I do. DNA replication happens at an extremely fast rate every time a cell divides, copying all 3.1 billion DNA nucleotides with virtually no mistakes. I hope you think this is as cool as I do, because it really just is. So I've now explained to you how cells copy and pass on their DNA when they divide and multiply. But what is DNA used for anyway? And how does it get used? Well, ultimately, our DNA's purpose is to store all of the information needed to assemble all of the proteins in our bodies. In essence, all of our physical characteristics. So how does that happen? How is DNA used to make proteins? Well, here's my cute little summary. To make a specific protein, the information found in DNA that codes for that protein is first extracted and used to make a complementary strand of RNA called mRNA, which stands for messenger RNA. This process of converting DNA to RNA is called transcription. And what in the heck does the word transcription mean? Well, according to dictionary.com, the word transcribe means to make an exact copy of. If I, for example, were transcribing a document, that would mean that I would be making an exact word-for-word copy of it in the same language as the original. So the process we're addressing here, transcription, is taking a segment of DNA whose language is written in nucleotides and making a complementary exact copy of it in mRNA, whose language is also written in nucleotides. This process happens inside the cell's nucleus. Now at this point, the mRNA strand exits the cell's nucleus and is subjected to a process called translation. The word translation means to convert from one language into another. Thus, this process converts the information found in RNA, written in the language of nucleotides, into an actual peptide or protein, which is written in the language of amino acids. The intermediary between mRNA and a peptide or protein is something called transfer RNA, or tRNA. Now, I don't want you guys to worry about all the details of this process yet. I'll explain those momentarily. I just want you guys to see the overview. When a cell needs to make a specific protein, the information in the DNA that codes for that protein is extracted and transcribed to form a corresponding molecule of mRNA. That mRNA's information is then translated using tRNA as an intermediary to ultimately form a peptide or protein. This is the process by which the information found in our DNA is actually used to form flesh and blood. So how does transcription actually happen? Well, when a specific protein needs to be made, an assembly of various enzymes unwinds the needed segment of DNA from within our chromosome that codes for that particular protein. A specific DNA segment that codes for a particular protein is called a gene. The enzyme assembly then makes a complementary mRNA strand from that DNA. Now here's another cool video from the DNA Learning Center that shows how that occurs.
What you are about to see is DNA's most extraordinary secret, how a simple code is turned into flesh and blood. It begins with a bundle of factors assembling at the start of a gene. A gene is simply a length of DNA instructions stretching away to the left. The assembled factors trigger the first phase of the process, reading off the information that will be needed to make the protein. Everything is ready to roll. 3, 2, 1, go. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the two strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to copy the A's, C's, T's, and G's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related building block known as U. You are watching this process called transcription in real time. It's happening right now in almost every cell in your body.
Translation is the process by which messenger RNA, or mRNA, is converted into an actual protein. The way this works is absolutely amazing. Here's an example mRNA strand shown. This mRNA molecule is shuttled into an assembly of proteins called a ribosome. This ribosome then reads the mRNA's nucleotide sequence 3 nucleotides at a time. The example mRNA strand I've shown here has every 3 nucleotides colorized differently just to help visualize this a little bit more clearly. So what happens next? Well, the ribosome assembly starts bringing in these molecules called transfer RNA, or tRNA. Here's a picture of a tRNA molecule. You'll notice that the molecule drawn flat kind of looks conveniently like the letter T. To me, the 3D computer model shown here kind of looks like a laser gun from a 1960s science fiction motion picture. In any event, the butt of this pistol right here has a 3 nucleotide sequence called an anticodon. The muzzle of the pistol over here has a cytidine-cytidine-adenosine, or CCA, tip that gets attached to a different amino acid for each different tRNA. So here's a math question. How many ways can you vary a 3 nucleotide long sequence when each position in that sequence can only come from one of 4 different nucleotides? Adenosine, guanosine, cytidine, and uridine. Do you know the answer? Of course you do. You guessed it. The answer is 4 times 4 times 4, which equals 64. So what this number means is that there are actually 64 different kinds of tRNA. one for each possible 3 nucleotide long anticodon sequence. Each of these 64 different tRNAs carries, attached to the muzzle of its gun, a different amino acid. Well, sort of. You might remember from chapter 23 that there are really only 20 different most common amino acids used in protein synthesis. As I'm sure you're aware, the number 20 is much smaller than the number 64. So how can there be 64 different kinds of tRNAs, each attached to a different amino acid, if there are only 20 amino acids to pick from? The answer is simple: redundancy. So here's how that actually works. How the nucleotide by nucleotide language found in mRNA is converted into amino acid language. And I really can't speak highly enough of the people who made these discoveries: Marshall Nierenberg, Har Gobind Khorana, and Robert Hawley. Once again, following DNA transcription, the newly formed mRNA is brought into the ribosome complex and read 3 nucleotides at a time. The ribosome then ushers in a tRNA whose anticodon is complementary to that 3 nucleotide sequence in the mRNA. So let's look at our mRNA here. This example that I've kind of stolen from the book, you'll notice that the first 3 nucleotide long sequence, which is called a codon, is AGC. Now, of all the 64 different tRNAs that exist floating around in the cell near the ribosome during translation, there's only one tRNA whose anticodon is complementary to AGC. And that would be the mRNA, or the tRNA whose anticodon is UCG. That particular tRNA will have attached to its muzzle a specific corresponding amino acid. And which amino acid is that? Well, to figure that out, we just have to look at this table. So here I have AGC in my memory RNA. If I look up at the table, I'm going to look at the first nucleotide, A, and then I'm going to go to G. G is over here in the third most column. Then I'm going to go down to the letter C, which is right here. You'll notice that that amino acid is serine. What that means is that as this mRNA is being dragged down the ribosome, a tRNA whose anticodon is complementary to this, that is, the tRNA will have an anticodon that says UCG, will have a serine dangling off of the end of it. That serine, once that tRNA is brought into place, will be released from the tRNA and added to the growing peptide chain. The next 3 nucleotide long sequence in our mRNA is CUU. What amino acid does that correspond to? I look at the table. C, U, and then I go down here, U. It's leucine. You see that? What that means is that there is a tRNA that would be complementary to this. In other words, the tRNA's anticodon would say GAA on it, and it would have dangling off of the muzzle of it a leucine molecule that would be added next onto the polypeptide chain. This would continue down the sequence until we got to the last 3 nucleotide long sequence here, GAA. What amino acid does GAA correspond with? Well, I look at the table. G, I look at the column A, and then I go here to A. It corresponds to glutamic acid. So once again, there is a tRNA that would fit into a GAA hole. That is, it would have an anticodon complementary to it that would read CUU. And that tRNA would have dangling off of the other end of it a glutamic acid residue that would be added to the growing polypeptide chain. That is how the mRNA language, which is written in nucleotides, is translated into actual protein language, which is written in amino acids. Now, you might remember me talking earlier about redundancy, and it is exemplified in this example. For example, you can see that the sequence UUU codes for phenylalanine. UUC also codes for phenylalanine. You can see that UCU codes for serine, as does UCC, UCA, and UCG. So you see there's a lot of redundancy built into this system. Even though there are 64 different tRNA molecules, several of those, according to this table, actually do the exact same thing. That is, they'll have the same amino acid dangling off of them, or in the case of UAA or UAG or UGA, they will code for stop codons. What does that mean? It tells the ribosome that that is where it should stop making the protein. At that juncture, the ribosome will disassemble, release the mRNA into solution, and also release the peptide or protein. The protein then goes into the cytoplasm from where it can be further modified or used as needed. I hope that all makes sense. I'm going to show you a little bit more information momentarily that might help clarify it even more. To recap, when our cell needs to make a protein, what it does is it opens up the segment of DNA found in the chromosome stored in its nucleus that code for that specific protein. It then brings in a collection of enzymes that take that segment of DNA, which is called a gene, and makes a complementary segment of RNA that corresponds with that segment of DNA. This process is called transcription because we're going from one language into an exact copy or complementary copy of the same language, which is nucleotide language. Messenger RNA is then taken outside of the nucleus and it's put into a collection of enzymatic machinery called the ribosome. And the ribosome then brings in these molecules called tRNA or transfer RNA. Each transfer RNA reads the mRNA strand 3 nucleotides at a time. When the tRNA finds a hole that it fits in, the amino acid that that particular tRNA has dangling off of it is added to the growing peptide chain. That continues until a codon in the mRNA called a stop codon is found. Once that occurred, the peptide or protein is released into the cytoplasm, after which it can be further modified or used according to the cell's needs. The ribosome complex disassembles and the mRNA is taken into solution to be digested and recycled. Now I realize that this figure we're looking at is a little more complex than is probably necessary, much like our IRS tax laws. But I hope you get the idea. And as you probably anticipated, here's one last video from the DNA Learning Center that shows how translation occurs.
When the RNA copy is complete, it snakes out into the outer part of the cell. Then, in a dazzling display of choreography, all the components of a molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. Each transfer molecule carries a 3-letter code that is matched with the RNA in the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecules. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second. And as a result, our muscles, brain, and all the vital organs in our body receive the oxygen they need.
So are you guys ready for some problems? Good. Let's take a look. I want you to answer me this question. What oligopeptide is coded for by by the following stretch of mRNA. Now I should give you a hint, you're going to have to use the table that I showed you a few slides ago. Now I want you guys to answer this question. What was the nucleotide sequence in the original DNA strand from which this mRNA strand was formed? Now, as I did in our previous presentation, I am not going to give you the answer to this question, either of the questions. Sorry. But I will give them to you in class. Now, although I could address numerous subjects discussed in our book, including DNA sequencing, genetic engineering, and gene therapy, time limitations constrain me to only highlighting one more topic. The topic that I've chosen to teach you about is the polymerase chain reaction, also known as PCR. PCR is a technique invented by Carrie B. Mullis in 1983, which allows scientists to make billions of copies of DNA in a relatively short time. So why in the crap would you want billions of copies of DNA? Well, there are numerous applications of this, but one of the most obvious is criminal investigation. Let's say that you're a detective at a crime scene where you find a small tissue sample that might belong to the perpetrator. You probably want to make multiple copies of the DNA in this sample for analysis so that you don't run out of DNA halfway through the investigation, right? How do you do that? The answer is PCR. So here's how PCR works. We take a sample of DNA, which might be large or small, and we heat it to 95 degrees Celsius, which causes the two strands in the DNA to separate. We then cool these two strands to 54 degrees Celsius and stir them with short complementary pieces of DNA called primers, which are shown here as little cute red and yellow boxes. Now these primers base pair or anneal to the opposing ends of the two DNA strands. We now warm these strands in the presence of a heat-stable DNA synthesizing enzyme and stir the mixture in a solution that's filled with an excess of free-floating deoxyadenosine, guanosine, cytidine, and thymidine. The enzyme, called a thermophilic DNA polymerase, then uses this supply of nucleotides to read down the original DNA strands and synthesize two complementary strands. We now heat the mixture up again and break apart and separate all four of these strands from each other. The process is now repeated, ultimately giving 8 complementary strands, which are then subjected again to the same cycle. Now each cycle amplifies the number of DNA strand copies exponentially. Thus it takes about an hour to complete 30 cycles, at which point the DNA has been copied a billion-fold. Now we have more than enough DNA, hopefully, for extensive analysis of our crime scene tissue sample. Now, one humorous thing about TV shows is the fact that they often portray scientific techniques quite inaccurately, and sometimes very ignorantly. Though I don't have time or the legal permission needed to share the following clip with you here, I did find a funny TV clip on YouTube that makes PCR look humorously much faster than it really is. Now, if you're bored enough to actually watch it, then you can find it here, the following HTML. Now that concludes our Chapter 28 presentation on nucleic acid chemistry. I hope it's been as fun for you as it has for me, and for my colon. So until next time, I wish you a good and happy DNA.